for work. So, what did you want to talk about? Like CRISPR, if you don't mind me asking you, because I noticed just from talking to many evolutionists, especially I think it was Jackson Weed who said it, because I just want to clear something up in regards to alleles, because I feel like you don't make that contradiction, but when you actually define an allele, would you define an allele as one of two or more versions of a particular DNA position, or just two or more versions of a gene?
It can be either, depending on what you're talking about.
Because,
I mean, for instance, when you talk about a SNP, you talk about SNP alleles, right? But that's a single nucleotide. So in that instance, it is one single position. But often you'll talk about alleles of genes, in which case it's the entire gene that's being discussed. So it depends on what you're actually talking about.
Okay, so it can be defined as a version of a gene, but we know that a gene can contain hundreds to thousands of DNA positions, right?
Yes.
Okay, so kind of like we talked about earlier, then I can define it. Like if you define it in the terms of just a gene unit, then generating allelic diversity by, say, mutating just one gene, say per mutational event, it would produce little diversity, right? Because I noticed, like Jackson Weed, a lot of the people I've spoken with, when they're defining it in that term,
you would have 140 something novel alleles per zygote. That's
okay, but I'm just saying if I were to define it as a single genomic position, say independent of its relationship to an actual gene, then obviously enormous allelic diversity can be generated by mutation. But I mean, we talked about before that obviously the diversity itself doesn't need to arise just from mutation. It can arise from recombination, gene conversion.
Well, actually, though, the problem is that if you're defining the allele as a single nucleotide, then you can't actually generate diversity. You can generate recombinatorial diversity, but you can't create any actual novelty in terms of sequence novelty.
Okay, so the combination of genes, say on the genome, can change due to the DNA arrangements, but you're saying based on, because obviously that sort of genetic variation, it is important. I mean, even
in terms of genetic diversity, yes, that's true. The combination of alleles present within a population is very important, hence change in allele frequency over time.
Okay, so you're saying genes on separate chromosomes, obviously we agree they're not linked, because not all genes
generally they're not linked.
Okay, so genes that are farther away, say from each other, they're more likely to actually be separated in homologous recombination, right? But when genes are on the same chromosome but very far apart, they're still going to sort independently, say due to crossing over the homologous recombination.
This is something that I was talking to Sam about when he was in here, which is that I think that when you hear the phrase genetic linkage, you think about the fact that you have two alleles that are on the same chromosome, that they're on the same piece of material, on the same molecule, right? But actually, the linkage that's being referred to is not the physical connection between alleles, it's the statistical correlation of inheritance for the genes, not the allele.
Okay.
Right.
Yeah.
So the fact that you have recombination between two alleles, so you have two alleles that had been on one strand, right, and now they're on separate strands, that doesn't break the linkage. They're still linked. The genes are still linked. Even if there's recombination, they're still linked.
Okay, so before we get to that,
no, I want to make sure you understand that because it's really important.
Well, does that all fall in line with Mendel's principle of independent assortment? Because obviously that's stating that alleles of different genes will segregate.
Mendel's principle of independent,
not always the case, right?
Mendel's independent assortment, properly speaking, only applies to the chromosomes themselves.
Okay. But these alleles of certain genes are inherited together. So they don't, in that case, they do not appear to undergo independent assortment at all.
Yes, right? when you have linkages, yeah, when you have a statistical linkage between genes, as is quite common when they're on the same chromosome, especially when they're quite close to each other, then they don't have the appearance of independent assortment. They in fact have a statistical correlation, statistical dependence in terms of inheritance.
Okay, so what you're saying is when two genes are close together, say on the same chromosome, and correct me if I'm wrong, they do not assort independently, therefore they're said to be linked, but genes located on different chromosomes, they assort independently, but they'll have a recombination frequency of about 50%, while the linked genes have a recombination frequency that would be somewhere.
I wouldn't say that. I wouldn't use the word recombination when you're talking about separate chromosomes. Recombination, I would reserve to discussion of, well, recombination, which is within a chromosome, right? I mean,
okay, just for people listening, so technically recombination occurs, and you might have a better definition than me, but I'm saying like when two molecules of DNA exchange pieces, say, of their genetic material with each other.
Right.
And the most notable examples of recombination takes place during meiosis.
Yeah.
And these homologous chromosomes,
that's right.
And swap segments of DNA, right?
So I wouldn't. If you're talking about two separate chromosomes entirely, I wouldn't talk about recombination.
Okay, so but you're saying, in regards to my model, because just for anyone listening too, like if they're confused here, we would say that before the fall and after the fall. The two different copies of, say, Adam and Eve's DNA would have been reshuffled at least two processes,
four copies.
Recombination, we're talking about the gene conversion, but this would make each offspring unique, leading to diversity, say, within the human race.
Right. So
obviously, just one sec. But to claim, say, that the millions of DNA differences that separate each person in regards to our model being functional front-loaded DNA differences and based on the allelic gene position definition that I explained earlier, you don't believe that enormous potential for variation is there?
I'll explain why. So okay, let me ask you a question though first. How many differences do you think there are on average between two people?
Between two people, there would be I've got calculations here. How many would you say? Obviously millions.
I think the number is about 20 million on average.
Yeah, okay, so tens of millions of DNA differences would separate any two people, but we're still 99% similar, 99.5% similar. Okay,
so now here's second follow-up question. Do you know approximately how many unique variants have been found?
So you're talking about single nucleotide variants? How many?
Yeah. If I'm remembering the number, it's over 350 something million. Now, obviously, you don't expect any one person to have all of them, right?
No. Well, because I would say that a lot of the diversity and the differences were due to mutations, say, after the fall. But I would say about 95% of them are front-loaded DNA differences from the start.
Okay. Now, I'm going somewhere with this, right? So you need Adam and Eve in combination to have more than that.
Okay, well,
and they need to have between them 350 plus unique variants on three chromosomes as compared to the fourth.
Okay, okay. I just want to follow you. So, with where you're going, so we agreed that human chromosomes differ by millions of single nucleotide polymorphisms, right, between and within individuals.
Well, no, what I'm saying is that if you want 95% of the currently existing variation to have been front loaded, and then also for there to have been a lot of variation that has since become fixed, right? That it's been lost, especially when you had the flood event, right? Then you need Adam and Eve to be carrying themselves. Let's just round it up to 400 million variants.
Because you're looking at these DNA differences that you would say are due to mutation. I'm saying
No, I'm not saying they're due to mutation. We front load all of them, right? They're all front loaded, but they have to be front loaded into Adam and Eve, right? So you need to front load 400 million mutations into Adam and Eve.
Okay, yeah. Keep going.
Okay, so that's a lot. But the variation that you need to see, the phenotypic variation that you need to see in humans from 400 million differences isn't that great.
Okay,
But you also need bats. You have the bat kind, right, that has to be taken on the ark and then spread out and become all the different species of bat. They need a lot more than 400 variants to account for all of that diversity. They may need as much as 10 times as much, but 10 times as much is bigger than their genome is.
Okay,
That's
so you're
the problem.
Okay, so let's say the ancestors to all modern day bats, okay? Let's say they possessed millions of heterozygous nuclear DNA sites. New species of these
They
bats
do now. They do now have millions.
Pardon?
They have the same number of variants that we do in proportion to their genome size. But because you're starting with so humans, you're going to start with 2 humans, and you're going to end up with all the modern humans, but all of the modern humans don't have much genetic variance they don't have much phenotypic variance.
Low diversity, low allele, right? Is what you're saying?
I'm saying that humans would need less of this preloaded heterozygosity to explain the current humans, as opposed to if you're trying to explain all of the varieties of bat that we currently see with a single pair of bats, right? Getting all of the modern humans from a pair of humans is a lot easier than getting all of the modern bats from a pair of bats, because there's more diversity in the entire clade of bats than the entire clade of humans.
But what so originally at the creation, you would have created, obviously God would have created these original created, say, let's use a bat kind, for example. It would have just not 2, it would have been a population of them.
But how many bats did Noah take?
Okay, let's say Noah took, let's say he took 2 of each, okay?
2 of each species of bat? Or 2 bats?
No, let's say he took 2 of each. So whatever the bat kind is, he would have taken 2 or 7. Right. So that could be a maximum of 14. So let's even say he took 14.
I guarantee bats are not considered clean by the Jews.
Yeah, 2 or 7, they're not clean animals. The
so
7 animals that you would take were specifically because you would be taking along sacrificial animals.
Yeah, and by the way, sacrificial animals to be sacrificed, so they did not enter the gene pool. But anyway, the bats were definitely not clean, so only 2 bats.
Okay, so let's say 2 bats. Let's say that some DNA variants were lost, before the flood or after the flood.
You would have needed to start with about 4 billion or so variants.
Okay, now
that's bigger than their genome is.
But if we actually estimate 50 million DNA pre-existing differences created for bats in general, and based on the processes of recombination and gene conversion, you're saying that these new nuclear chromosome combinations will not arise each generation? Like, do you think that's a problem?
actually, no. What I'm saying is that recombining the order of these genes isn't going to change the phenotype in any great way.
Okay. I get what you're now. If we're going to produce new species of these bats, obviously these traits and these nuclear DNA combinations, they're going to have to be isolated. So whether
Yes.
it's small population sizes, inbreeding, migration, this can lead to the founding of new, more homozygous populations.
That's absolutely true. That's absolutely true. I'm not arguing with that. What I'm arguing with is that you would need to have preloaded into these genomes more variation than the genomes can hold to explain all of the daughter species, right? It is literally impossible.
Overload? Are you saying there'd be genome overload?
I'm saying that you have the bat genome, which I don't know exactly how big the bat genome is. Let's say it's 3 gigabase, right? I'm saying that you would need to have 4 or 5 or 10 gigabase worth of variants in a 3 gigabase genome.
Okay, so when it comes to the bat kind, why do you think that they are front loaded with this with the same amount of heterozygous DNA, nuclear DNA sites as, say, a population of cats or dogs or humans?
Well, I mean, that's your claim.
Yeah, okay, but based on the nuclear DNA differences that we see today, how many nuclear DNA differences separate, say, bats and humans, or bats and cats?
I wouldn't know the exact number. What I do know is that the number of variants that separate any 2 individual bats of the same species is going to be about the same by percentage of the genome as what it is in humans. And the number of total variants that we see in bats is going to be, again, about the same as the number of total variants we see in humans by percent of the population, or percent of the genome size. It's pretty late here, so I'm getting a little word choice is not as great. And what all of that means is that to get the variants seen in all of the different species of bats from just 2 that you're starting with, the amount of total variation that you would need to start with has to be greater than what you have now. Because if not, you can't have a loss of heterozygosity to form these independent species, right?
Have we found all the genes that would actually code for traits? Because obviously
we don't need to. It's in the DNA, right?
Yes, it's in the DNA,
right? So if we've sequenced the entire genome, which we have, and we know how much variation there is now, which we do, then we know that the amount that had to have been there at the start had to be much greater than what it is now, because that's what your model requires. And to get that would require an amount of variation greater than the size of the genome, which is impossible.
Well, so kind of like analogous to a computer. There's only a certain amount of information that can be expressed in that computer before it's hard drive overload, right? You're saying that the information systems of these bats, based on the single nucleotide variants, and compared to our model of pre-existing heterozygosity, you're saying that the genomes of these bats would be almost overexpression, is what you're saying.
An easier analogy would be that you're trying to squeeze 10 liters of water into 5 1-liter glasses.
Yeah, something like that.
Obviously, we know that our genome or our DNA system is a highly compressed genetic information system. So a lot of this information, a lot of these traits come about by decompressing this compressed information, because not all the information is being emphasized.
But your entire argument rests on the loss of heterozygosity in the formation of species, that you have front-loaded heterozygosity that is lost, right? That means that you have to start with heterozygosity that is lost. You have to have that initial heterozygosity greater than the current heterozygosity, otherwise your model is wrong.
and you're right, because as these more homozygous groups grow to larger and larger sizes, they will eventually be recognized as new species. But the original created kinds of, say, bats, how do you, are you comparing bats of what we know today versus?
What I'm saying is, if you're going to say that all modern bats are descended from a single pair of bats that came off of Noah's Ark, then those bats, and that's one, and that also all of the genetic diversity, or at least a significant portion of the genetic diversity of all of those species of bats, was already present. This is the critical bit. Was already present in the genomes of the bats that came off Noah's Ark, the pair. That means that the amount of genetic diversity in the genomes of that pair of bats then must logically have been greater than the total genetic diversity of all of the species of bats that exist now. Because otherwise, you would have had to have produced that novelty in the intervening period. And recombination cannot do it, because recombination does not produce huge variance in terms of phenotype.
Now, are you saying that the amazing compression ability of, say, the genome or even every nucleotide will not account, say, for the need of this larger genome, of this overexpression that you're referring to?
Yes, it absolutely will not. That's not how genetics work.
Now, so are you saying that based on recombination, gene conversion, because obviously for us, the heterozygosity, the millions of differences in these DNA sequences of the chromosome pairs.
I'm sorry, I just got to cut you off, though. The recombination and gene conversion. Firstly, gene conversion statistically is a reduction of heterozygosity, not an increase, right? Because gene conversion is biased towards the shorter allele in terms of multi-base conversions, and towards the GC allele in terms of single base conversions. So it's biased towards the same result every time it occurs. Additionally, it requires that there be
Okay, it's not a huge, I mean, gene conversion and recombination are still, like, in organisms, say, that possess two versions of their DNA sequences. Obviously, we would agree that a hybrid of the two versions is then passed on to the offspring. But the process of forming this hybrid DNA, that involves copying large chunks from each chromosome, as well as copying small parts from each chromosome. So gene conversion is the latter of that. It's almost a faster way of producing that combination of, it's almost like shuffling a deck of cards, at least in regards to recombination. So you're going to get limited potential for variation.
You have misunderstood the way gene conversion works.
Are you saying the definition that I just provided was an inaccurate definition of gene conversion?
Gene conversion is based on repair mechanisms. It's homology directed, and is biased towards copying the GC allele, right? So that means that if you have two different alleles on the homologous chromosomes, and one is an AT allele and the other is a GC allele, that the GC allele is the one that will be replaced, right? So that you'll have GC on both. So additionally, you're not going to replace a variant that is right next to another variant, because it requires very large areas of homology for gene conversion to occur in the first place. All is basically saying that gene conversion is only making your problem worse, not better.
Okay, so let's take this slow. So obviously when chunks of chromosomes are swapped, okay, this is what we refer to as recombination, just to keep it simple. So we agree to that. Now, a chunk of, say, the first member of the chromosome pair, let's just say if we use, I don't know, chromosome 1, for example, that might be swapped, say, with a segment of the second member of the chromosome 1 pair. But when tiny sections, or say even individual base pairs, are then swapped, that is the process we would
That's the
say
thing, is
is
they're
gene
not swapped.
conversion.
They're not swapped. That is your misunderstanding here. In gene conversion, there is not a swapping of material. It is a repair process.
Okay, and I want us to keep going with the situation, but just because I don't want to stop with the point you brought up earlier in regard to the overexpression, have we actually sequenced, say, all the compressed information in DNA?
Yeah, so
At this point, in order to
people like to talk about the compression of information in DNA, and really, inevitably, they're talking about stuff that they don't understand. You have several semi-independent processes that are happening simultaneously on the same sequences, but they're not storing more information than they look like they're storing. You've got these base pairs, and that's what you've got. Okay, so that's the amount of material you have that you can change. You can't change any more than that. You have to be able to change more than that to get the level of variety needed to get all modern species of what creationists would consider a kind from a single pair of progenitor animals. Ergo, the whole thing's impossible.
Well, when I think of the genome, I look at it as, a hierarchical system. Like, for example, it'll function at 3 levels. So the DNA, deoxyribonucleic acid molecule itself, also the DNA to RNA to protein complex, right, that makes up chromatin, and the 3-dimensional arrangement of chromosomes in the nucleus. But at all 3 of these levels, DNA can function in ways that are independent of its exact nucleotide sequence. Would I be correct in saying that?
That would depend somewhat on what you mean.
I guess I would say, like, because obviously we've had in-depth discussions on, ENCODE, the results ENCODE has revealed, the functions of non-protein coding regions. Would you say you underestimate the actual functionality of the genome itself, or do you look at it?
No, I think I've got a fairly accurate gauge on the functionality of the genome itself.
Do you find that, would you be able to say confidently that in the future, because it seems like more and more papers are coming out about the, significant functions that obviously portions of our DNA are doing and providing. Would you say that in the future that we're going to find more and more of these important functions, or do you think we're going to find more and more that a lot of these non-protein coding regions are just evolutionary leftovers? A lot of them are functionally dead. Like, how would you?
Here's my expectation. My expectation is that we found past tense 5%. I'm not going to use exact numbers here because it doesn't matter. 5% that had a certain type of function, right? And then we found a new type of function in 2.5%, right? And then we found another new type of function in about 1.25%, right? And I expect a pattern of roughly that type to continue on for some time, wherein you will find increasingly obscure forms of functionality in increasingly small numbers. That's the pattern that we've had.
Okay. So I know we touched on this before, but when ENCODE projects, they examined one, kind of like the percentages you were just saying, when they examined 1% of the non-coding DNA of, say, the human genome for possible function, and then they found, I think this is where you disagree, that at least 80% of the non-coding DNA was transcribed into RNA, which I'm saying is suggesting function. that would mean
Okay.
that the non-coding regions of the genomes correlate with biological complexity even better.
So here's
than
the question I
protein
need you to.
coding
Here's the
regions.
question I
I'm
need
saying
you to
there's
answer.
well, just 1 second, because obviously with the I just want to say that, you know, in my mind, and what this evidence suggests to me is that the non-coding regions do indeed carry significant information, say probably for regulatory activity. So that's an underestimation of the actual functionality of the genome, I think, on your part. Go ahead.
Well, only if true,
Yeah, no,
which you can't demonstrate, and all of the evidence suggests is false. But here's the question that I would need you to answer, which is that what percent of an entirely random stretch of DNA would you expect to find transcribed?
So an actual stretch of DNA, what's the percentage of that I would expect to be transcribed?
If it was entirely and completely random.
I'm not too sure how much of it I would expect, like percentage-wise, to actually be transcribed versus, say, spurious transcription. Is that kind of what you're getting at?
Well, I mean, you would agree that if you had a stretch of entirely random, well, let me ask this question first then. If you had a stretch of entirely random DNA and you found that it was transcribed, some of it was transcribed, you would agree that that would constitute spurious transcription, right?
Obviously spurious transcription exists, and it obviously they knew of spurious transcription even before ENCODE. Did this testing, but
I haven't made my point clear. I feel I haven't made my point clear.
Yeah,
Let's say I design a computer program to generate an entirely random sequence of DNA, and I produce this entirely random sequence of DNA that we know is entirely random because I made it that way, completely random. And we put this entirely random sequence of DNA into some eukaryotic cells, and then we sequence their transcripts, and we find transcripts from this entirely random stretch of DNA that I designed to be entirely random. You agree that that must be spurious transcription because we know absolutely that it's completely and totally random, right?
like in your theoretical random segment that you just
Yes.
Okay, sure. Yeah. Well, and just to clarify before you continue with your point, from my understanding, ENCODE did test every nucleotide and discovered that it does have biochemical activity. So the answer to your question, I would say, would be 98%.
98% what?
I think I'm saying 98% functional, but obviously
Of the completely random DNA?
now.
You're saying that completely random DNA would be 98% functional?
I would say that based on ENCODE's revelation that every nucleotide has biochemical activity, because if only 50% of the genome is conserved,
Well,
and evolutionists, from my understanding, predicted 80% or more, why would that be?
well, I'm sorry. This is really confusing now, because now you're saying that you think that almost all of an entirely random stretch of DNA is functional, and that also 80% of the DNA that we test that's non-coding is functional. Well, I agree that any possible definition of the word functional that includes entirely random stretches of DNA, if you're using any definition of functional that includes that, then sure, most of the genome is functional. It must be. If most of random DNA is functional, I can't imagine how it couldn't be true. But that's a silly definition of functional. It's a definition of functional that not only does no one use in biology, but it's completely useless.
I understand, but what's your definition of?
Has an effect on phenotype.
Okay, so it has an effect on phenotype. Now, ENCODE looked at what was biochemically active, right? So that was
Does not necessarily have an effect on phenotype.
Okay.
If
Now,
it's not biochemical, let me say this though. If it's not biochemically active, it's not going to have an effect on phenotype, right? So if your expectation is that this region will have an effect on phenotype, then finding that it's got a biochemical function is a good place to start. But it's not where you stop. You now have to actually demonstrate the actual function.
And I understand, but because obviously, you know that I'm saying that the trajectory of discovery I'm saying is favoring genome-wide functionality. I'm not saying I can tell you what every single function is.
Now, the problem is that your understanding of that trajectory is wrong, because as I said, there's an asymptote here that is at a certain level, right? Because we are getting significantly diminishing returns in terms of the amount of the genome that can have each newly discovered type of functionality attributed to it.
Well, from my understanding, and correct me if I'm wrong here, from a lot of sources I've read from, and from even a lot of quotes from ENCODE scientists and researchers themselves, it seems like most evolutionists who are not actually heavily invested, say, in this creation evolution debate, they seem to embrace the ENCODE result and to embrace what I'm saying here. But it seems like a lot of atheistic evolutionists, say, who are really invested in this creation evolution debate, it seems like they're heavily disputing the reality of what ENCODE has discovered. Because when I say, I'm almost done, because when I say non-coding DNA is probably involved in regulatory functions, right? Obviously, when and how fast to make proteins, embryonic development, that's a huge one that we can get into, the timings of protein manufacturer, and so on and so forth. It seems like, from the everything I'm reading, they're not in dispute to that. But it seems like you are in dispute to my claim that the non-coding DNA is involved in those things. Go ahead. What do you think about that?
Well, firstly, I'm certainly not disputing the fact that some percentage of non-coding DNA is involved in regulatory functionality. That's been known since the 60s.
How much would you say is involved in that?
Somewhere between 3 and 10%.
Okay, so 3 and 10%. And how much percentage of our genome is protein coding?
5 to 8.
Okay, so say 5 to 8%. And you're saying how much is non-protein coding? Sorry, 3 to 5% or something?
Functionally non-protein coding.
Yeah. So how much?
Yeah. Let's say I'll just put raw numbers on it. Functionality, I expect somewhere between 8 and 20.
Of the whole genome, 8 and 20.
Yeah.
Okay. Do you believe? Okay. ENCODE. Did they look at the portions of the genome involved exclusively in development? Because we know such DNA would only have a function then. Like, did they or didn't they?
So that's a weird way of phrasing the question.
Well, because what I'm saying is the reality is that we have only just. I don't know if.
Okay. Let me rephrase my statement.
Yep.
So, if your argument is that the some region of the genome will only have biochemical functionality during some particular stage of development, then the fact that ENCODE found anything or didn't find anything for that region means exactly nothing.
Did ENCODE predict, though, I don't have the quotes with me, but from what I read, they predicted that 100% of the DNA would have function. But are you saying that their definition of function is the same definition of function that you're providing? Like, it's not...
No, their definition of function is useless. And that is the consensus of the bioinformatics community.
Are you disputing the definition of function that the ENCODE researchers and scientists used? Okay,
yes.
so you're in
consensus
with what they've said.
Yes, the consensus of the bioinformatics community and the biological and chemical community at large is that the ENCODE project shot themselves in the foot by using a definition of functionality that was, to put it somewhat mildly, somewhat insane. You cannot come up with a definition of functionality that is as broad as they did. Because their definition of functionality includes things that could not possibly be functional.
Could not
possibly be functional by any reasonable definition of functionality.
Amongst people like yourself, scientists like yourselves, and the ENCODE scientists and researchers. You're saying there's a conflict there, but and you're saying that most evolutionary scientists are on your side, but
not most evolutionary scientists. I'm sorry, not most evolutionary scientists. Most people who are involved in any aspect of biological research, biochemical research, bioinformatics research, or any of the associated fields. ENCODE, and specifically, by the way, ENCODE was a massive project that produced an amazing amount of high-quality data, right? The data and the quality of the data and the quality of the people, for the most part, who produce that data is not in dispute. The problem is that many of the leads felt it necessary for the purposes of their own careers to somewhat overreach in terms of their conclusions. And if you look into the backgrounds of these people, you can see that there's some tension there with a lot of the people they interact with. This is not something that is my view. This is a common view from people who are familiar with the topic. All right.
Okay, so when they say, like the ENCODE project, when they say that they found, very convincing evidence that the genome is pervasively transcribed such that the majority of its bases can be found in primary transcripts, including non-protein coding transcripts, so obviously, because it seems like from what I've looked at, most of the evolutionary community has almost abandoned even the term junk DNA, and they seem to be siding with ENCODE.
Yeah, almost no one has actually abandoned. You could come up with maybe one or two fairly decently big names who would want to say that junk DNA is a bad idea and that there's widespread functionality, but the overwhelming majority of the people who are actually involved in the sort of bioinformatics work that's relevant to this would agree that. Sorry about that. Would agree with the sort of reasonably conventional view of junk DNA. Would be hard pressed. Let me put it this way: you would be very hard pressed to come up with a panel discussion that was, say, 5 members by 5 members of pro versus con on junk DNA, because the pro junk DNA side you could fill up right away, and the people who think that it's pervasively functional, who the last 3 would be a challenge.
Okay, so it would be your assertion and your, I guess, belief that some non-coding DNA is proving to be functional.
No, that's absolutely true.
The minority of the non-coding DNA, right? Like, you don't expect that there's going to be any more, because it seems like
I'm sorry. I expect that there will be more found, but for every new category of functionality, right, I expect that category of functionality to apply to a smaller and smaller percentage of the DNA. Okay, and
what would you say are the stats of, say, people from the scientific community who, say, support ENCODE and their findings and their definition of, say, function, versus individuals from the community, the scientific community, that are, say, siding with
5% maybe? I don't know.
So you're saying,
I don't know. I really don't know.
But you would say that some evolutionary scientists, you're obviously saying it's a minority, would be, say, in agreement with obviously some of the ENCODE scientists, researchers, and what I'm saying as well?
Yes, sure. Some percentage would.
Okay, but
I'm sure that some percentage would say that HIV is a made up thing, right? Like, you get some weird things if you you get people to talk outside of their subspecialty.
But if I look at things like, quotes even by evolutionary scientists that say like most of the genome that we used to think was there for spacer turns out to be doing stuff, as in like most of our genome is at least not idle, and the fact that a lot of these scientists do believe that portions of the genome are involved exclusively function-wise in development. That means that they expect a larger percentage to be revealed to be functional in that stage. Would you say you disagree with all of that, or how much percentage do you believe?
I know a lot of people who are involved in developmental studies.
Yeah.
Right. And they have similar views of ENCODE that I do. Right. I mean, it's just not a plausible position to hold.
Well, from my understanding, and correct me if I'm wrong, or maybe I'm understanding this, but expressed RNA sequences have actually been analyzed, I think, in organ donors, aborted fetal tissue, and embryonic stem cells. Obviously, I don't agree with the latter of those things, but nevertheless, this data has served to increase the known functionality and complexity of the human genome. But obviously, yep, go ahead.
I don't think you could possibly say that the fact that you find transcripts in aborted tissue that can't tell you that there's functionality, it can only tell you that there's expression.
Okay, when it comes to let's just say your experimental results then on the just human DNA sequences that we're agreeing demonstrate biochemical evidence for function.
No,
I'm saying that's just the beginning of our understanding.
No, I'm sorry, you can't say it that way.
Okay, what would be your
you can say biochemical activity.
Well, isn't that what I just said?
But well, you said biochemical functionality.
Okay, but I think the most important point of what I'm saying, and that can be taken from all this, is that the trajectory of these results, and correct me if I'm wrong here, it seems like we've watched the scientific community go from, say, claiming high levels of non-function in the early 2000s to claiming evidence for nearly pervasive function even a decade later. And I understand that not all scientists are saying this, but it seems at least like the ENCODE researchers themselves, who don't really have a bias in this, that's at least what they're saying. So why don't we do more experiment?
So firstly, the ENCODE researchers have a bias towards the ENCODE project.
Okay, so you would say that
should be pretty obvious, right?
Okay, so you're saying that they're trying to upsell the results of it, because obviously you're saying that they have a bias to the project itself. They want to make the project look.
That's exactly right. Yeah.
Okay.
The other thing is that I would say that since the 70s, you have had the overwhelming majority of biologists have a view of junk DNA that the majority of the genome, varying percentages depending on who you would ask and at what specific time, but the majority certainly was not functional, with some percentage of the community saying that there was pervasive function, and that now you have the same thing, right? I don't think that that's actually changed in any real radical way. What you do have is you have data sets that are available that can be interpreted in a naive way to give a conclusion that could support pervasive functionality if you ignore the fact that the types of data in use do not actually demonstrate any type of functionality, which is a real problem.
Okay, from my understanding, though, and I'm not saying you're twisting facts, but I wouldn't want it to be that way, because from my understanding, ENCODE didn't actually assume evolution. They were actually neutral. And from articles that I've read, mainly there's one called, I think, The Evolution of Television Sets, where these evolutionists are actually criticizing them for being neutral. So they neither had an evolutionary bias nor any other bias, say, for example, a creationist bias. So the fact that they're neutral and the fact
I don't care about that.
But well, you should care because obviously you're saying that they had a huge bias to their project. But from my understanding, it seems like it's a hidden secret. That we are seeing this pervasive function, but to the evolutionary community, the ones that have a high investment in it, they're criticizing these ENCODE scientists for being neutral. So that's my understanding of it. I think that's true.
Well, no, I'm sorry. Firstly, I don't care whether or not ENCODE built in evolutionary assumptions into their modeling or not. That doesn't actually matter to me. What?
Well, do you think they had a bias, or do you agree with me that they were neutral? They did their work.
I think that they were
perspective.
I think that they were neutral in the production of their data, and in the presentation of their data, they were biased towards selling the project.
Okay.
The problem that I have though with ENCODE, because I'm not particularly offended by aggressive selling of results. Within reason.
Well, do you believe that they were results though? Like it seems like you're disputing.
Yes, no. So exactly. This is the second clause of what I was saying.
Okay,
is that they decided to aggressively sell their results by selecting a definition of functionality that allowed them to include a much broader swath of the genome than what any reasonable analysis would allow. Okay, so in other words, the thing that I take issue with is them using a silly, I'm sorry, but it's the right word here, a silly definition of functionality to set their thresholding and to set their selective criterion.
Now, would the ENCODE researchers themselves, the scientists, would they also agree with your opinion that their definition of function, whether it's biochemically active, is silly? Would they agree with you on that, or would they dispute you on that?
Well, I think that the lead would dispute me on that. I think that a lot of the individual researchers would probably agree with me, but I would have to talk to each of them individually, and I think there's like 200 names on the ENCODE consortium. I don't remember, but I mean. But the fact that, but hold on, I just want to say this one more thing, is that the fact that even if you say that every single member of the ENCODE consortium would disagree with me on my assessment of the definition of functionality, I would still be in agreement with the overwhelming majority, again, not of the evolutionary community, but of the scientific community as a whole. ENCODE, in terms of that definition, is a massive statistical outlier.
Let's talk more like if we're looking at, say, certain parts of the genome, because we know exons and introns are both transcribed, right? But the latter are then cut out and the former spliced together, I think, in alternative ways. But is there a debate on whether or not these introns are just passive spacers, or that they actually play important roles? Because wasn't it assumed that those sites that tend to be close to the affected exons were not really serving any purpose, but now that the results we're seeing, they actually suggest regulatory elements that are, say, deeper into introns. They're a little more appreciated. There's a little more action to them. Because if they're encoding a majority of the small RNAs, like how would you feel? Like, is there a debate on whether or not they're just passive spacers, or there's more complexity to it?
Yeah, so this is an interesting example of exactly the kind of thing I was talking about, where you have new types of functionality discovered in increasingly small percentages of the genome.
Okay, but this is a new discovery that there is a debate on whether or not
whether or not it can happen, or whether or not it happens as a rule.
Okay, I get what you're saying. So as a rule, but so the non-protein coding RNAs, say from introns, I'm saying from what I've seen that they influence gene expression by, say, modifying chromatin. The complex combination of DNA, RNAs, and proteins that make up chromosomes. That seems pretty important, and that's just more of what scientists.
It's possible. It's possible that some small percentage does that.
How much percentage would you say?
Well, it would be within the 8 to 20.
Well, if junk DNA, or if the areas of those non-protein coding regions are, say, telling proteins when and how to produce their project or product, doesn't that suggest function to you? And
yes,
the
it
paper,
suggests that they would be part of that 8 to 20% that is functional.
it seems like, and I know I've used the words DNA language so many times, it just seems like you really don't understand.
Let me rephrase this number. I'm sorry. Let me rephrase this number in a way. I think that there's approximately 10% of the genome that we have very strong evidence for functionality for,
Okay.
and another 2 to 3% that we have reasonable to somewhat weak evidence of functionality for, and the remaining 7% to get us to 20 is maybe, I don't know.
Okay,
Probably not, but it could be.
Okay, but
right?
why are 50% of, say, pseudogenes, which I know are used as genetic markers to prove universal common ancestry through phylogenetic trees and charts, how come they're now proving to be functional and yet they're in the 80% junk areas? Doesn't that just show
if they're
more and more how much
if they're functional, they're not in the 80% of junk by definition. However, and this is something that you do a lot that you really need to watch, is that you take a category, right? And then you name a subset of that category that has function, and then you say that the entire category has function.
Okay, so you're saying that in that category, there's a, let's say, a segment of function, but then you're saying the remainder of the category is non-functional, is what you're saying.
I'm saying that the remainder of the category has no evidence of functionality.
Okay. So no evidence of functionality. Now, I've never said that I claim all of the category is functional, but I'm just saying that there is a strong trajectory, and I don't think you know for
but the thing is that there's not actually a strong trajectory of that, right?
Oh, there's not. Okay.
So the trajectory that we see is that for every
disagree with you on that, though, just real quick before you continue.
Is what?
Would the ENCODE scientists and researchers disagree with that statement you just said?
I have no idea what the ENCODE scientists would think about that statement, but I can tell you that the overwhelming majority of the biological community at large would agree with that statement.
What's the percentage of the biological scientific community that agrees with you, say, over the scientific? You said 5%. Do you stand by that?
That would side with ENCODE?
That would side with you. Oh, no, yeah, that would side with ENCODE. But 5%?
Could be. Yeah, I mean, I don't have the numbers, so I don't actually know. But that's my impression of the field. Right. So I'm vaguely involved in a number of different areas. And again, you have to understand that my background is cancer, right? And so when you're doing cancer research, evolutionary biology can be informative in some instances, but really what you care about is what fights cancer, right? So, people are not terribly likely in the cancer community to turn down an avenue of research because it doesn't conform to evolutionary biology. I've never met somebody who would. I'll put it that way. So I'm engaged in with the bioinformatics community, with the biochemical community, with the oncology community, with all of these areas that are not, strictly speaking, under any kind of evolutionary constraint in terms of the work that they do. And of all the people I've talked to, people tend to look at ENCODE and think, wow, that was a lot of work they did. Why did they come to those conclusions? That's weird.
Well,
that's the consensus from my experience. Okay, and these are not evolutionary biologists.
I guess my question would be, and tell me if you think this is a valid question. It seems like a lot of junk areas have actually been overturned and proven to have function, say, against the prediction that I think was before 2001. Why would that be, and why wouldn't you believe that even more areas assumed to be junk will be overturned in the future if we've agreed that, say, in regards to introns, in regards to other, debatable areas of function in those areas. How come you don't believe that there's going to be even more revelations in regards to unknown functions in the future? Like, it seems like you don't have a very confident.
What? Confident what?
It doesn't seem like you have a strong confidence in, research, and because I'm saying that if so many areas have been overturned from these junk regions, why don't you believe in the future there's going to be more?
So it's always interesting when you find something that goes against the general rule, right? But you have to understand that finding any one thing is going to represent a thousandth of a percent of the genome, right? It's vanishingly small. And even if you apply it to everything that matches certain characteristics of this thing that you found, it's still going to add very, very, very little. Now, the real point that I'd like to make, though, is that if you say that in 1970 the estimation of functionality is let's say I don't know, 10 plus or minus 5, right? And then in 1990 it's, I'm making these numbers up again, that it's 12 plus or minus 2, right? And then in 2010 it's 12.5 plus or minus 0.15, right? You could point to, well, it's going up all the time, except that it's not, because it's always been within the initial error bars, and the error bars are getting smaller all the time.
Okay, so to clarify, and just answer the question quickly, based on what you think now, function-wise, our genome has, say, in a percentage, versus what we will find in the future, based on what we know has been overturned in those areas, what do you think the max, like if you were to put a number on it, almost a prediction that you can put on paper, what do you believe the max percentage of functionality? Because obviously I'm saying that there's
sir, it cannot be higher than 20%.
Okay. Now, if I'm saying that embryonic development may need over 50% of the genome as manufacturing tools, what would that, in the DNA, okay, what would be that step-by-step process, those manufacturing construction tools? What would those be during the embryonic development? And do you believe that those have been studied?
Sorry.
Like, one last part to that, because would you agree that the dynamic use of, say, DNA sequence during development is very different than the vast majority of DNA sequence that is used as an adult? Like, would you say that there's a huge dynamic in usage between
there are absolutely differences in expression profiles during development versus during maturity. However, if you're going to say that 50% of the genome is required for development, you're mistaken.
Why would I be mistaken, though? Okay, can you give me the step-by-step, or can anybody give me a step-by? Because from my understanding, they know very little on a genomic level based on those manufacturing and construction tools, because the number of possible scenarios in this developmental stage in regards to DNA sequences.
So there's no strong
positive function is actually proving to be enormous, right?
There's no strong evidence. There's no strong evidence that any significant percentage of the transcripts found during any of the stages have any evidence of actual functionality to them, especially given what we know about things that actually do have function. They don't seem to match any of that, and also they seem to match things that we would predict of stuff that has no function.
Okay, but do you know what
It is
DNA
generally
step by
speaking.
step during that? Because I think that's the most important question. Like, what does the DNA do step by step during the embryonic development? Because I'm saying 50% of the
It's like,
genome is needed. So what does
it's like this. It's like this. If 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 you ask me, do I know step by step how to build a house, and I say no, that doesn't mean I can't say that rain falling on the exposed foundation right after they dig the thing out isn't functional, right? I can still say that.
Okay, in regards to the house, then building of a house, would you say that a lot of construction tools, manufacturing tools, and workers are needed during that process?
Yeah, and I would say that it fits comfortably within that 20% max that I've already
Okay,
specified.
okay. But after that house is built, what happens to a lot of those tools? Obviously, most of the workers and anything involved in the manufacturing and construction of the house. What happens to a lot of that once the house is built?
Well, a lot of it is still utilized in other ways.
What about the
A lot
workers?
of it's inactive.
I'm talking about in regards to the house, the analogy of a house, because you brought up a house. What happens to those workers who were heavily involved in the construction of the house? What happens when the house is built? Like the house.
In the analogy of the house, it's more like a log cabin where you expect that the at least some of the people involved in building it are going to live in the thing afterwards.
All of them? All of them?
No, not all of them, but some of them. Some of
Percentage
them
of-
definitely.
What's
Right?
the percentage of them that would move into the house that they built?
Come on. I
Okay.
don't have specific numbers
Okay,
on
let's
that.
be real.
Yes,
So a
but
lot of these,
there are definitely, let me put it this way. There are definitely genes that are involved in development that are not expressed after development.
so they're silenced.
Silence would carry with it certain implications that I don't necessarily want to use. Some of them are silenced, yes. Others of them are simply no longer transcribed. There's a difference there that we don't need to get into.
But they still had a function at some point, because I'm saying experimentally testing a DNA sequence.
No, yeah, this is sure. They were functional at some point.
Well, to just in each of these unique windows of time that I've been talking about, in which, say, sections of the DNA are actually used and then silenced, would be enormous.
Yeah.
Now,
So the nice thing is the nice thing about that is that there are a number of these things that we have found sort of accidentally because of genetic variations that have caused problems that we've then isolated and identified and been able to pick these things out. And what we found is that they follow a number of patterns that everything else follows, right? And that in turn allows us to search through the genome for other things that also follow those patterns, and they've allowed us to do significant annotations.
Well, wouldn't the silencing, though, let's say if there is silencing,
no, it does not affect it.
Okay,
no, because what we can do is,
wouldn't it look junk?
let me explain this.
Wouldn't that DNA look junk, though? Just yes or no?
No. The reason is that when there's a random variation that causes a defect in development, right, that allows us to isolate the bit of the genome that is defective in that animal, and it allows us to then track that back to that same section that is not defective in other animals, right? And we can then look at it after development, right? And we can see what it looks like after development. We can see what it looks like before and during development, right? And we can take those characteristics that we see before, during, and after development for these genes that we know to be involved in development, right? And we see that those match all of the other genes that we're familiar with to a very high degree. And what that tells us is that it's very unlikely that the stuff we're looking for is going to be significantly different from everything else that we're familiar with. Yes, it may be silenced immature organisms, but we know what silenced functional genes look like, and they don't look like junk DNA. They
Well, based
don't
on
look like the 80.
so much junk DNA has been overturned since, say, 2001. Obviously,
But it's false to say that. Hold on, I'm sorry, but I'm sorry. You keep saying that. You keep saying that, but you're wrong.
I don't
You
believe
keep saying that so much. The quote unquote.
You have with the actual scientists doing the work and coming up with results because of the bias. And you're saying that you do understand what the DNA does in embryonic development, but it's a fact that it's a big mystery. I think there's a lot of fact twisting here.
It's not a fact, actually.
No, because you can't give me a step-by-step process of what DNA does and governs during that embryonic development. And you're underestimating what it does.
Things are transcribed.
Well, we
transcription factors bind. Things are transcribed. I mean, developing organisms are extremely complicated, right?
Yes,
so
I agree.
Here's the real, I think, failure of comprehension that comes in with this is that the mechanisms that are involved, we understand. What we don't have is a complete mechanistic map of how all of these pieces ultimately work together. But we know what the pieces themselves look like. Okay.
Okay, but if it's not, okay, let's say if it's not DNA, governing embryonic development, then what else would it actually be? Because would you agree with this? And let me just go over this, because obviously we know in that short 9-month window of time that obviously represents human embryonic development, a single cell turns into a fully formed baby that we know contains hundreds of cell types that have to execute an unimaginable number of cellular tasks. So surely we would agree, I'd imagine, that the developing baby would have to call upon enormous amounts and variety of DNA code to execute this developmental program. Would you agree with that?
I think it fits comfortably within 20%.
Okay, so you believe that fits comfortably within 20%?
Honestly, I suspect that it fits comfortably within 12, but that's neither here nor there.
Okay, so if it's not DNA governing embryonic development, so you would say that
it is DNA.
Yeah, so it is DNA governing all of that. Now, would you say that based on the comprehensive knockout experiments that we have done to determine the function of DNA, say in mice, because we know we haven't done it in humans for ethical reasons, would you say that's enough for you to conclude, kind of like in our last discussion, that it is only, say, 20% involved in that phase that I've described? Go ahead.
That's a big part of it. Bioinformatics is a big part of it. Just basic understanding of biochemistry is part of it. There are several ways that you can get to this, and they all everything sort of coalesces to the same numbers. And that, the honest truth of it is that you're asking me here to sort of distill together, my entire collection of experiences, my professional experience, my educational experience, the personal relationships that I have with all of these people in all of these different fields who are actually actively involved in many cases in the forefront of the areas of research that they do, and deliver it to you in a way that is easily digestible in a short period of time, when honestly it's not my specific subfield. Right, like if you want to learn about this, there are universities you can apply to.
Because I believe a lot of the data is being cherry picked. So if you want to send me some articles and papers on the step-by-step process of what DNA does and how it governs
I would suggest that you look into C. elegans.
So, because I don't believe
you're going to want to look into C. elegans.
Well, why would ENCODE scientists and researchers believe that a lot of the genetic regions they're testing are going to be involved in this? And apparently they're not aware of all these.
I have no idea why you would ask me to try and mind read these people.
Have you read much of what they've said in regards to the findings, in regards to?
I read the original ENCODE article. I read several of the follow-on articles. I've read many a blog post, both for and against.
Okay,
And-
let's just say then, because I don't want to go around in circles. I know we'll agree to disagree here, which is fine. I think we had a good discussion on ENCODE, on junk DNA, but for our prediction that it involves heavily in regards to these non-coding regions, I believe, and correct me if I'm wrong, because since you're an evolutionist, evolution would say that there must be a lot of non-functionality. But in our model, we predict functionality for most of the genome. I think we've gathered that from now. So I'm saying that ENCODE project have suggested, and I know we agree to disagree, that at least 80% of the human genome is functional. Now I'm saying they're preliminary. I agree with that. But I think the trajectory is clear, especially in regards to our discussion here on embryonic development, that most non-coding DNA in the human genome has to be functional and serve at least in a regulatory capacity. Now, in regards to the pre-existing DNA differences, tens of millions of DNA differences, I think it all comes to this. This is my point all along, is that if they are functional front-loaded DNA differences, and I think if there's not, they're all not being expressed. There's a lot of compression involved, and the fact that we speak very little of the DNA language. I mean, back in the 30 years ago, it was one gene, one protein, one function. Now we know of protein moonlighting. I mean, it's like a multifunctional tool. RNA was underappreciated. Now we know there's an immense amount of functions involved in RNA. So I'm saying that there is a strong correlation to these facts and our model of pre-existing DNA differences. Go ahead.
Yeah, I mean, all I would say is that you're basing that on, and no offense is meant here, but it's based on a very limited understanding of the field as a whole.
And that's fine. I just want to say that it's been fun. Ken Hovind just uploaded Ken Hovind versus Aaron Raw debate part 1. I'd suggest watching that. Before I go to bed, I got to get up early. I want to go watch that. I think it'll be fun, probably funny. Did you know they're going back and forth with they're doing a video by video debate. So Aaron Raw did one, and now Kent just uploaded his response. So I'm going to go watch that and have some fun.
I imagine that Aaron Raw is engaging in that for very much the same reason that I do this.
Well, think about it. Aaron Raw in his round 1 had like 50,000 views. And obviously, I know you strongly disagree with creation, but if you look at his other videos and say the last 2 months, I mean, that was like quadruple the normal views he gets, right? So I think it's going to be fun regardless of if you're an evolutionist or a creationist, right?
Sure.
Come on, you agree with that. The stat in regards to the amount of views, but you just admitted you enjoy engaging in it, right?
Oh yeah, personally, sure. I don't watch a lot of the videos from a lot of these from a lot of the bigger names, except for like PZ, but that's just because I've actually been in several hangouts with them. So.
Oh, PZ Myers, yeah. Okay, guys, we'll do this again soon. Maybe we can do it on my channel next time. I think that'd be fun. But yeah, it looked like that turned into a little longer than half an hour, but you need that for these types of discussions, right?
Yep.
Yeah, sure.
Okay. Thanks, Athena. Thanks, Crispr. Peace.
Yeah.
Yep. Good night. Anyways,
with that, I think I'm gonna have to go ahead and go as well.
Yeah, I'm gonna end it.
This is fairly good talk.
So. Yep. Hang on. Let me end this. Hold on.